Okay, good, perfect. So everyone, welcome for the second session of lightning talks. We have 3 again tonight, so 5 minutes each. Now you know the rule, and then you can go to the posters to ask the questions. So I think tonight we have 3 PhD students. If I'm wrong, I'll just correct me later. So the first one will be Jacqueline Boccacino from University of São Paulo in Brazil. She's going to talk to us about stemness and early embryo development. So you're on, Jacqueline. Good luck.
Thank you. So hello, I'm Jacqueline Boccacino from the Laboratory of Neurobiology and Stem Cells at the University of São Paulo, Brazil. I'm an undergraduate student working with genomics and bioinformatics. And today I will present our work in which we evaluated the roles of stress-inducible protein 1, STI1, in early development and embryonic stem cells. So protein homeostasis or proteostasis plays an important role in pluripotency maintenance because stem cells require fine control of proliferation, self-renewal, and pluripotency itself. And STI1 is a cochaperone protein that interacts with several clients and participates actively in proteostasis. It is known that STI1 is essential during early development because the lack of this cochaperone notably impairs the embryogenesis of early murine embryos, although the molecular mechanisms behind this are still unknown. Therefore, we hypothesized that STI1 may regulate the biology of embryonic stem cells. So to investigate this possibility, we analyzed publicly available single-cell RNA-seq data of early murine embryo cells ranging from the zygote to the late blastocyst stage. We also developed an in vitro model deriving embryonic stem cells from the inner cell mass of early murine embryos that were transgenic for the STIP1 gene that encodes STI1, resulting in embryonic stem cells with either normal, lower, or higher levels of STI1. We first focused on the bioinformatics analysis, so we clustered cells according to their developmental phases, and then we assessed STIP1 expression in each of these stages. We were able to observe that STIP1 expression is increased in specific stages of development from the 4-cell to the intermediate blastocyst stage. Importantly, STIP1 is co-expressed with several pluripotency genes. So here, SOX2, PO5F1, and ANOG correspond to the main core of pluripotency regulators, whereas the remainder genes correspond to an extended core. Afterwards, we performed some trajectory analysis within the blastocyst stage, which is when the inner cell mass develops, and we found that STIP1 expression reaches its peak during the intermediate blastocyst stage, along with several pluripotency genes. Finally, we computed the correlation between STIP1 and all genes during the blastocyst phase, and we verified that STIP1 is positively correlated with many pluripotency genes, especially ANOG, PO5F1, KLF4, and SOX2. To gain further insight into which processes may be regulated by STI1 in embryonic stem cells, we developed that in vitro model that I mentioned before, and we performed a bulk RNA-seq on those embryonic stem cells with different STI1 levels. Our results indicate that cell cycle and also processes related to stem cell biology are differentially regulated upon distinct STI1 expressions. So the striking findings provided by both bulk and single-cell RNA-seq data analysis prompted us to investigate STI1 functions through functional experiments. So at the transcriptional level in our model, we verified that STI1 impacts the expression of some pluripotency genes, such as SOX2, KLF4, and ANOG. Then we verified the protein expression of OCT4 and SOX2, observing that STI1 affects the expression of pluripotency markers at the protein level as well. Finally, beyond pluripotency, we were also interested in ascertaining whether STI1 could impact other aspects of embryonic stem cells. So we verified the implications of STI1 in the proliferation and DNA damage of embryonic stem cells with different levels of STI1. And we found that those embryonic stem cells with elevated expression of STI1 are more proliferative and exhibit less DNA damage than those with lower expression of this cochaperone. So taken together, our results indicate that STI1 is an important orchestrator of early development in a stage-specific manner, being a pivotal molecule that can regulate critical aspects of mouse embryonic stem cells. So thank you so much for attending my talk, and feel free to ask any questions and to discuss my presentation in the poster session that follows. Thank you.
Thanks a lot, Jacqueline. Perfect.